Hello students, this is Inheritance and Variation Part 10. In this slide, you are going to study the pleiotropy in detail. So what is pleiotropy? When a single gene controls two or more different traits. Okay, when a single gene controls the two or more than two different traits, it is called as pleiotropic gene. And the phenomenon of this process is known as pleiotropy or pleiotropism. So the phenotypic ratio in this pleiotropism is 1:2 instead of 3:1 because of the death of the recessive homozygote. So the disease that you are going to study in the pleiotropism is sickle cell anemia and is caused by a gene that is known as HBS. You are going to study it. So normal or healthy gene, it contains HBA. HBA is dominant one. The carriers, the heterozygotes, HBA and HBS, these are the heterozygotes. They show signs of mild anemia as their RBCs become sickle shaped, that is half moon shaped, only under abnormal condition. And because of the sickle shaped type of structure, they cannot carry the more amount of oxygen inside them. So in the pleiotropy, when a single gene controls two or more different traits, it is called as pleiotropic gene, and this phenomenon is called as pleiotropy or pleiotropism. And the ratio, as I have mentioned earlier, it is 2:1 instead of 3:1 because of the death of homozygotes in the pleiotropism. According to Mendel's principle of unit or character, one gene or factor controls one character. Okay, but sometimes the single gene produces two related or unrelated phenotypic expressions, and that you are going to study in the pleiotropism. So we are going to study the structure of RBCs in detail in the next slide. You can observe the point mutation which causes change in the structure of RBC as we have studied. I have mentioned earlier, I have explained in detail what is mutation, and we are going to study that how the mutation uh, occurs in the phenotypic way, how we can observe phenotypically the mutation, the change in the structure. Okay, so that you can observe structure of RBC. Now this is the normal structure of RBC. These are the normal red blood cells. Now you can observe in the next slide, in next image, that RBC, now this is cytoplasm containing hemoglobin. So these are the normal circular biconcave discs with no nucleus, and these RBCs or red blood cells, they carry oxygen. Now these are the normal type of RBCs, but when there is point mutation, when there is change in the structure of red blood cells, now how they look in the sickle cell RBCs, the structures get changed. Okay, now you can observe the difference between the two RBCs. Now this is the normal red blood cell, while this is the sickle cell. Okay, so this area you can observe, this is a complete circular disc. So this normal RBC can carry more amount of oxygen than the sickle cell, which is half moon shaped red blood cell. So this type of RBC, it cannot absorb or carry that amount of oxygen which this normal RBC carries. So that we are going to study in detail, that how the point mutation, what is the point mutation in the next slide. Now let's talk about what is point mutation in detail. Now this is the normal beta polypeptide chain. You can observe these circular numbers, okay, blue colored. These are nothing but amino acids. These amino acids, they form polypeptide chain. Now this chain is different than this one because you can observe the sixth number. Now from 1 to 8 all are same, while this sixth one is yellow in color in case of sickle cell anemia. Here we are talking about the change, okay, a point mutation. So in the sickle cell anemia, the amino acid positioned at the sixth position, that is glutamic acid, it get changed or it get replaced by a different kind of amino acid, that is valine, and this change is known as point mutation. So in normal human body, our hemoglobin or protein part contains 2 alpha or 2 beta polypeptide chains in the normal human body. In sickle cell anemia, in one of the beta polypeptide chain, there is point mutation. So here we are talking about, now this is a normal beta polypeptide chain, while this is the abnormal one where the point mutation has occurred. So this point mutation causes sickle cell anemia, and it is an autosomal recessive disorder. So we are going to talk about in detail what is sickle cell anemia in the next slide. So before going to the direct part of the sickle cell anemia, let's try to understand this flow chart first. So you'll be able to understand sickle cell anemia in detail, what kind of disease get occurred because of this point mutation. So in the previous slide, we talked about point mutation, how it occurs in the amino acid chain at the sixth position of amino acid. So due to that point mutation, there is change in this gene at the sixth position. Because of this point mutation, the structure of normal hemoglobin get changed. The beta chain get changed, and because of that, the structure of the normal hemoglobin it get changed, and because of that, the structure of red blood cells get changed. That is, the normal circular disc shaped RBCs or red blood cells get converted into half moon type of structure. So that is the structure alteration in the RBCs. Because of this change in the red blood cells, due to this, the oxygen carrying capacities of the RBCs get decreased. Okay, the surface area get decreased. And of course the capacity get decreased, and because of that, the person's physical activities get affected and the efficiency get decreased. So before talking about the chart of sickle cell anemia, it is very essential that you must understand the meanings of these terms. That is, what is HBS, what is HBA, what if HBA and HBA get expressed together, what if HBA and HBS get expressed together, and what if HBS and HBS get expressed together. So if you understand these terms so very clearly, you can write the answer and you can understand the sickle cell anemia in detail. So if a person carries HBS type of allele, it is the recessive gene and which is responsible for the sickle cell anemia disease. See, try to understand, only one HBS is present, so it is recessive, so it will not get expressed, but that person will be a carrier. Okay. Well, in case of HbA, the normal hemoglobin, that person will carry normal hemoglobin and normal red blood cells. If the person carries HbA and HbA, normal hemoglobin that person will be having and normal shape of red blood cells. If a person carries HbA and HbS, then this person carries 50% normal hemoglobin and 50% defective RBCs, as HbS is also getting expressed, and HbS is the recessive gene which is responsible for the sickle cell anemia disease. Okay, so there are 50% chances of the defective RBCs they can get expressed in that person. So this person is resistant to malaria. This is another advantage of having or carrying the HbS along with HbA. They don't have normal RBCs, so don't carry good amount of oxygen, so they get tired due to muscular activities. So HbA and HbS, if it's this type of genotype, if it is getting expressed, then that type of person will be having this type of uh, characteristics. Well, if a person if it carries HbS and HbS, then both the genes are defective. As you know, HbS is lethal one recessive gene and it carries sickle cell anemia disease, and both the genes they carry HbS and HbS information, so it is of course going to be very lethal. Both the alleles from the sickle cell RBCs, and normally these individuals they don't survive, and because of that we have studied about the ratios. Okay, So that is, if the person carries HBS type of genes, then that person dies. It doesn't survive. So in this slide, you can observe the representation of pleiotropy. If the parents, now these are the phenotypes, sickle cell carrier. So if that person carries the gene, but it is not getting affected, okay, so its genotype will be HBA, HBS, HBA and HBS. If the parents, they carry the sickle cell anemia disease, then the gametes, they will get formed. The genotype will be HBA, HBS. Of course, the gametes, they will get formed HBA and HBS. Okay, and in the offsprings, these kind of chances are there. Genotype HBA and HBA can get expressed together. The phenotype uh, will be normal. If the genotype of one of the offsprings is HBA and HBS, HBA and HBS, these are the combinations of HBA and HBS, then these offsprings will be sickle cell carrier just like their parents. They will be having HBA and HBS, sickle cell carriers. They will also survive. While if the offspring, one of the offsprings, if that offspring will carry HBA, HBS and HBS, both the lethal genes, okay, so sickle cell anemic offspring will be there and that offspring will not survive, it will die. So in the sickle cell anemia, in this disease, sickle cell anemia is caused by a gene that is known as HBS. We have talked about it in detail in the earlier slide. So normal or healthy gene is HBA, which is the dominant one. And the carriers, that is heterozygotes, HBA and HBS, they show signs of mild anemia as their RBCs become sickle-shaped, half-moon shaped structure, and they have oxygen deficiency, but they don't get affected. They are just the carriers if they are having HBA and HBS type of genes. Okay, they are said to have sickle cell trait and are normal in the normal conditions. The homozygotes with the recessive gene, that is HBS, however, die because of the anemia as it is very lethal. So the homozygotes with the recessive gene, that is HBS, however, die of total anemia. Thus, the gene for sickle cell anemia is lethal in homozygous condition and it produces sickle cell trait in the heterozygous carrier. Now, this is the homozygotes trait, while these are the heterozygotes traits. They survive. These are the carriers, while sickle cell anemic offspring, it dies. So the two different expressions are produced by a single gene, that is HBS. It forms two different kinds of expressions, and a marriage between the two carriers will produce normal carriers. Now this is normal offspring, these two are carriers, and another is anemic. So the ratio is 1:2:1. Sickle cell anemic dies. These patients, they don't survive. These offsprings, they don't survive. So the normal ratio is 1:2. Okay, so the heterozygotes or the carriers can be identified by the microscopic examination of the blood. So that's all about the sickle cell anemia. So if the question is asked, you have to write these important points. These are the notes. Okay, and of course the chart is very essential. If you try to understand the previous slide, if you try to understand the genes or the genotypes, then it becomes very easy. There is no need to mug up all the things. It becomes very easy. All the things, the gamete formation and their combinations, which are indicated by the arrows, that you have to write in detail in your examination. So in the today's session, we have studied the definitions, alleles, multiple allelism, mutation, what is point mutation in detail. As far as your NEET is concerned, these points are very important. You have to remember the names of the amino acids and all. And of course, the diagrams are essential when you are, the charts are very essential when you are attempting the questions, whether it is on multiple allelism or whether it is on pleiotropism. But you have to remember the charts. I have explained the various examples, two examples, and of course codominance we have studied by taking another example, that is AB blood group. So number of points you can collect, you can write, and the points are very easy. So go through your books, prepare your notes, read the points. You can note down the points from the slide itself. The flow chart which I have provided, these are kinds of notes which the chart again the HBS, HPA, all the genes and the genotypes. So by remembering the charts and the flow charts, it becomes very easy to study. So hope you have uh, studied and it is understandable to you. So that's all for today's session. Thank you.